Hi, my name is Caroline Morin, and today I will be walking you through my tutorial to visualize axolotl gene expression during limb regeneration after forelimb amputation using R. The first step is to prepare the environment. We will be installing and uploading the R package, Tidyverse. This package is a large collection of other packages involved in data wrangling, visualization, and exploration, which we will use to create data plots. To access the data, we must go to the website axolomics.org. This was created by Stewart and Thompson Laboratories to upload supplemental axolotl data. Here we can retrieve the data we will use in R by going under the tab and clicking here, then pasting the link into R. Now we can upload the data directly into R using Download File to download the information from the URL given. File Show allows us to see the text file within the R environment. The uploaded data is still in a text file format. We need to convert it to a readable file. Using read_del_m to make the text file into a data file. After that, the data file now has extra information we want to filter out. Here, using Group By to select the desired data, Symbol was the labeled column with the genes, and the numbers as columns have the gene expression values. I also used Rename With to remove the X in front of the time points. We can now create a new data frame with all the specific genes we are interested in. Using Filter to filter the genes in Gene Count and grep_L to search for the specific matches allows us to capture each unique gene within the data, as well as making the data frame into a longer format using Pivot Longer. This is easier for us to graph with later on. We also need to make time a factor. Because the times listed are in both hours and days, we must tell R to keep them in this specific order, so they appear so on the graphs chronologically. To now visualize gene expression, we will use ggplot, which creates data visualizations, allowing us to better observe the data in our data frame. Here we are plotting all regenerative genes in our new dataset, Regen. This will output a line graph of all gene expression levels over the course of time they were measured in the dataset. This visualization shows us expression levels from all the genes within the family selected. The highest levels of expression are between 6 hours to 1 day, spiking around 12 hours, with an outlier from MMP12, showing maximal expression around 6,000 TMP. This increases the magnification of the rest of the genes, distorting our visual observation. To better observe the genes, we must remove MMP. To now plot the genes while excluding MMP12, we will use the same ggplot from above while using Doppler filter and Symbol to specify which gene to exclude from the graph. This now shows us a better representation of all the genes excluding MMP12 due to its magnitude of expression levels, and we can see the specific levels throughout. We will now plot just MMP genes, while still excluding MMP12. Using Doppler filter to filter the data in Regen, to search using grep to search for genes with MMP in their name, while also using Symbol to specify which gene we want to exclude, in this case MMP12. Here, on our graph, We can now more clearly examine the other MMP gene levels expressed throughout this time period, with a clear incline around day 1, starting at 6 hours and decreasing around 3 days. We can also see another spike around day 7. Because of its high levels, it is unproportionate to graph with the other genes involved. So we will plot MMP12 by itself, while using Doppler filter to filter in regen with symbol in to identify the value MMP12, confirm its presence, and then plot it by itself. Having MMP12 plotted, we can better observe its extreme increase and decrease in expression, jumping from 0 to 4,000 and then 6,000 over a span of 9 hours. It is the highest expressed gene throughout all the extracted regenerative genes in our data, indicating its significance in wound healing and blastema formation directly after amputation. We will now be plotting two gene families together, FGF and TGFB, because they are both growth factors, so it will be best to compare them together. We will still be using ggplot, and we will also be using Doppler filter to filter in the regen data frame, searching for the genes with grepl to find the genes FGF and TGF in their name. This will filter the data and only include the genes when plotted with ggplot. TGFBs are important during initial wound healing and blastema formation. FGFs are involved in later development of the limbs and patterning after blastema formation. However, this graph indicates two highly expressed genes, FGFBP1, green on the left, and TGFBPI, pink on the right, occurring at times opposite of their typical expression. To further analyze, we will separate the two genes with the highest expression and graph them together. To now further examine the expression levels of these genes, we will filter them from regen using the Doppler filter and symbol in to identify the genes that we want, and we will then plot with the ggplot. This plot allows us to closely examine the two genes of interest. FGFBP1 encodes for the FGF binding protein 1, which releases FGFs from the extracellular matrix, enhancing signaling and promoting cell proliferation, differentiation, and migration. FGFBP1 works with FGF signaling pathway to aid in regeneration, acting as a substitute for nerve signals. The TGFBI gene provides instructions to make TGFB1 protein, which is important for the preparation of limb development. It is expressed during the initial process phase and decreases as the limb starts to rebuild. TGFBI is also involved in the formation of the apical epidermal cap, which maintains the proliferation and survival of cells during regeneration. Both genes and their familiar pathways work in tandem, working to initiate healing and formation during the regenerative process. In this graph, these genes are reaching similar expression levels as they mirror each other. When one increases, the other decreases. However, further analysis is required to determine if these genes actually co-express. Moving on, we will now examine BMP genes. With the same script from before, we will also use Doppler filter to filter in regen data and searching with grep to find all BMP genes, while then plotting with ggplot. This graph is a great representation of the BMP genes. They are involved in the regeneration of bone and cartilage and other tissues, as well as the regulation of cell proliferation. These expression levels vary throughout, some maintaining their levels while others fluctuate. BMP5 stays consistently low, although it is important in the BMP signaling pathway, it is not highly expressed. However, BMP7 shows a variety of expression levels throughout. It has been identified through its role in blastema formation, and BMPR2 is a protein receptor 2 that is essential for the formation of new bone and cartilage. Its expression can be detected in the blastema with involvement in maintaining proximo-distal progression of regenerating limb. To now examine the WNT genes, we will use the exact same plot from before, with grep to filter for WNT genes within the regen data with filter, and then ggplot. The Wnt signaling pathway has significant influence throughout axolotl regeneration, promoting blastema formation, initiating limb regrowth, and cell proliferation. At 1 day, we can see an increase in many of the genes, while WNT5A and WNT5B have high levels from day 1 to day 21. This could correspond with the pathway's initiation in blastema formation and signaling of the limb regrowth. A complete limb regeneration takes about 40 to 50 days in juveniles, so we can see through this motion towards cell differentiation and limb growth is beginning to occur around this time, when the genes have increased in expression. Using the same code from above, we will also examine sonic hedgehog, or SHH gene, using Doppler filter in regen data and searching with grep to find SHH gene and plotting with ggplot. Sonic hedgehog has a crucial role in limb patterning during axolotl regeneration. As we can see, this gene is not expressed until day 5 and then peaks at day 10, then falling back to 0 at day 21. While it is being expressed, it is telling the cells within the blastema where to position to form posterior structures, or fingers. It is expressed only when it is required to give this information, which we can see in this graph. For our final plot, we will compare the highest expressed genes within BMP plot, WNT plot, and the sonic hedgehog gene. To examine the expression levels between these highly expressed genes, we will filter them out of regen and then plot, using the Doppler filter and symbol in to identify the specific genes listed: BMPR2, BMP7, WNT5A, WNT5B, and SHH, and then plotting with ggplot to show a line graph. This graph allows us to visually combine the highest expressed genes within these families. Because their signaling pathways are essential and contribute to the formation and patterning of the new limb. The influence these pathways have on each other is still being discovered, but evidence suggests coordinated interactions are occurring. While observing the graph, we can see the gradual increase in expression of these genes as regeneration is occurring. They each play a specific role, which is expressed when it is needed to be initiated. It is recorded that around day 10 post-amputation, the blastema is continuing to develop and grow, which aligns with the presence of these expressed genes. While all genes within this data are important in regeneration, it is also important to analyze specific genes which have similar interests. I hope that this manual gave a better representation of axolotl gene expression during limb regeneration. Thank you.